And I'm going to give a similar talk to a talk I gave 2 years ago, but we have some nice progress to report to as well. So the first half is going to be devoted to kind of basics of how to simulate data for population genomics, and then we're going to talk about like more recent developments. So population genomics generally is focused on this task of taking genomic data, genomic sequence data, and using it to make inference on past events that shaped the variation that we see. And there are, generally speaking, kind of two main themes that we're interested in. One is the demographic history, kind of the dynamics of history of populations to which these genomes belong. And the second aspect is natural selection, that also obviously affects patterns of variation. So the idea is that by understanding the processes that by which these kind of demographic events and natural selection affect a genomic variation, if we understand these processes, we can develop methods to do this inference. So we can infer various things of interest. The main problem is that I think for all population genomic inference is unlike in other fields of computational biology, we can't run an experiment to see if our methods work, right? Like in most computational biology, you develop a model or a method to try to computationally model your biological process, and then you can run an experiment to see how well it behaves. And here we don't have very efficient ways to model this forward process of evolution. So what people typically do is use simulations, and simulations have always been very central in validating methods and also kind of guiding the development of new methods. And as the data became more available, we have more complex data sets, and we want to be able to infer more sophisticated scenarios. Then we also have to make our simulators more complicated. So generally speaking, what people typically do is they have like an evolutionary scenario that they think is reasonable or natural, and then they use these forward models, which are pretty descriptive. I mean, they might not be completely accurate, but we can do pretty good simulations under very sophisticated models of evolution to create our genome sequences. Then we can apply our inference methods on this data to get the inferred history, and we can compare the simulated and the inferred history to see how well we did, and use this to evaluate methods. So that's kind of the common practice in population genomics. Now, one thing that makes this possible, or at least more efficient, for neutral simulations, so these are simulations where we assume there is no natural selection occurring, is that there are kind of two processes that are conditionally independent. One is the generation of the genetic genealogy, which we're going to talk about and was mentioned here before this arg, and the other is the mutation process along the arg. So if we're interested in neutral processes, then we can partition the simulation into two parts. If we simulate natural selection, that no longer applies, and we need to use more more time comprehensive approaches. So what is this genetic genealogy? So I think this was already mentioned, and probably going to see it maybe in other talks, so it's worth mentioning, spending a few minutes to explain, to at least get a good grasp of what this is. So a genealogy, you probably know, if this is you, then you go back in time, you have two parents, four grandparents, you trace your lineages going back in time, and it branches out exponentially, but then you start getting some of these branches end up joining, like this person here is your common ancestor, but through two different lines of, you don't see my mouse, right?
There's a laser pointer there.
This? Okay, all right. So this ancestor here is a common ancestor through kind of your mother and through your father independently. So because, again, we have finite number of people going back in time, so these lineages have to merge and cross at some point. And this genealogy goes back in time indefinitely, even before, like, this might be the human cutoff 200,000 years ago, it still goes back to kind of the dawn of evolution. So at least conceptually we can think of it as going back indefinitely. And when we now consider multiple individuals, then they're not genetic genealogies, then their genealogies start merging at some point, and you get this kind of nice lattice that connects them as you go back in time. And the idea is that this is kind of the thing that you're interested in. When you're analyzing data from multiple individuals, the best you can hope for is to understand what the kind of high-level structure of this joint genealogy is. So when you're doing, say, demography inference, you're trying to understand again, these events of when these splits in these genealogies occur, and there's good reason to believe why these are meaningful events that don't only pertain to these three individuals, because if you would take two individuals that are kind of from the same population, then the only difference in their genealogy is going to be in the recent populations. So these events are not really dependent on the individuals we took. They're probably going to be very similar if we took different individuals from the same, what we consider as a population. But none of this has to do with genetics. Until now, this is just the conceptual genealogy, tracing back ancestors back in time. How is this connected to genetic data? So if we kind of plot the genomes of these three individuals, and if we consider just a single site or a very narrow locus in these genomes, then this site here, if we look at its inheritance along these genealogies, then it is inherited by a single lineage going back in time for each individual. So whenever there is a branching point to two parents, then this site came from one of these parents, right? So if you trace, say, this green site from this individual B, it will go through a single one of the lineages that traces back in time. And the same from each of the other individuals, so you get one tree that is kind of embedded in this genealogy. And this is for a single site. Now, if you move to a nearby site, it's going to be a different tree. Potentially, it depends on how far you go, then it's going to be a different tree. And what you see here is that, like, some of these trees, like the green tree, its structure conforms with the kind of population tree of these three individuals, but the red tree has like a different structure, right? Because A and B find a common ancestor before they find a common ancestor in C. And these trees change, and the reason why these trees change is because of recombination, right? So whenever, if we move along the genome, whenever we encounter an ancestral recombination event, then the tree is going to change. This is this kind of sequential Markovian coalescence process that was mentioned earlier. So another way to to show the same thing is that we move along the genome. In each short region there is a single kind of tree, and the mutations are if we see a variations within this segment, for instance, B and C have a different variant than A, then this means that there was a mutation that explains this, right? So you can take the variation in the genome sequences here and explain them using this tree sequence here, right? So together, this is called the ancestral recombination graph. There are many ways to represent it. A common way is using kind of a tree sequence like this, where you just represent like the first tree and then the changes as you move along the genome. And on top of this, you can express the mutations that occur along branches of this tree. Oh, so all of that was just kind of conceptual theory, and one of kind of the first popular tools for simulating data under these models was this MS tool from Hudson. That's like a simulator that I used when I did a lot of my research in my postdoc, and that's what people did. And this simulated the coalescent width recombination that was mentioned before. And this is like a standard MS command line. I won't go through it, but it has a part where it tells you have to kind of specify the general settings, and then a lot of these commands here that where you specify the demographic history, like these EJ are the joins of populations as you go back in time. So in a way, what you do here is you specify some general settings, and then you specify the demographic history, like the population that merge and there is migration. It can simulate quite sophisticated scenarios, and then you will MS would simulate the coalescent width recombination going back in time. All of this was neutral simulations. Then another important step in progress, this is what people mostly use now in neutral simulations, MS prime. So there were quite a lot of contributions here that made this much better than MS. The main thing is that it's much more efficient, but it's also based on Python code, so it's a bit more robust. It's easier to code your simulation scenarios, not with these cryptic command lines, right? And another important feature is that it really relies on this tree sequence data structure. That's, first of all, that was the main source of its efficiency, but it's also a very useful framework for kind of communicating information about evolution. So you can actually store the sequences in these tree sequence data, and this is now a lot of inference is actually done on these tree sequences. So that's considered like the core way to represent data in many ways, not just in simulations. So if you kind of compare simulations with MS, this is what MS looks like, and with MS Prime, it's more like a Python code where you can write comments. It's much more friendly and makes simulations much more accessible and general. I'll get to, well, you don't see this highlight, but yeah, this is something I'll mention later. So all of this was simulating neutral evolution. There was no natural selection. And the main reason, again, why that is significantly more efficient is because you can separate the mutation part from simulating the actual genetic genealogy. So you first simulate the genetic genealogy just of the individuals you care about, and then you can kind of sprinkle the mutations on. When you simulate natural selection, you can't do that because the mutations themselves actually affect the way that the genealogy is going to progress. So you have to do forward simulations, pretty much. There are some methods that do good approximations with backward selection, but with backward simulation, but pretty much you have to use forward simulations. So there were some precursor simulators say 20 years ago, but SLIM, which was developed around 10 years ago, was really an important step forward. And again, making this much more accessible, where you can fairly easily describe your scenario of selection and using a Python script and then let it simulate data. It's more efficient. It's still much more time intensive than doing neutral simulations, but you can now simulate pretty complex scenarios of natural selection also and demography together using SLIM and generate pretty realistic simulations of genome sequences. So these are the two main what we call simulation engines today. We call them engines because they're general purpose, right? You can code different scenarios. They're designed to be engines in a way where you actually write code to generate your simulations under the scenario you care about. They're very general. They have a lot of support in terms of software, so a lot of very wide user base. So they're designed to be very, very general. So in a way, and I'm not saying that there's a lot of progress to be done there, but the main challenge now is not just to be able to simulate more complex scenarios. It's actually to be able to use these now pretty standardized engines in a way which is more robust and reusable, where people can actually where these simulations are really widely accessible, which is why people wanted to use simulations to begin with. Right? And the problem is that when you come to simulate data, you have to care about a lot of things having to do with kind of the genomic features. So there's a sequence length and mutation rates, recombination rates, the demographic model of the populations, the generation times, different things with selection, which regions are under selection, what selection regime you're going to use. There are a lot of things you have to set up, right? And first of all, there's a lot of ways to get this wrong. So you think you have something in mind, and then when you come to code in that translation, it's very error prone. And the second thing is that you want to be able, so I want a realistic simulation, right? But how do I set all of these things, right? I need to know the literature, what is the best version, and how to maybe combine things from different places. This becomes very complicated. So for instance, demographic model, right, I would want to take a demographic model that is published in the literature. So for human data, one of the most popular models is this Gutenkunst et al. Model from 2009, right? So you go to their paper and they have this nice conceptual image of the demography with three populations, Chinese, European, and African, and you have the image and you also have a table with actual values, right? So this is a good example. I mean, this is a good example where the data is pretty accessible. You can pretty much get the parameters from this table and plug them into your code. There are always conversion issues like the scale. What is the actual scale you need to use? There are a lot of issues. A lot of places, even here where it's pretty clean, there are a lot of places where you can get this wrong. And you think that you would encode this and you would run your simulation and you make an error, and then you actually run a simulation that doesn't simulate under this model. So it's a real big problem. So for instance, this would be the code you would write to simulate this model. This is actually the code that was, I think, published with the MS Prime software, right? To show, right, I can take a published demography and encode it nicely like this. This was written by the people who wrote the code for MS Prime. These are people who are experts in demography and reading these things and writing the code. Right. And something like 4 years later, maybe I'll get to that. Okay, so put a pin in that. We'll get back to that point. So around 2018, there was kind of the field. This is an idea, I think, that was kind of in the air for a long time, because people knew, a lot of people were using simulations, right, and coding simulations for their own studies, but everybody was coding their own simulations that were not reused. And that's a problem. First of all, there's no reason to code kind of simulations for the same scenario again and again. And that's one problem, right? We want to be able to use other people's code. We want to be able to combine different features. And we want also to have better quality control, right? So that you code your simulation, somebody checks it, and then you can reuse it like a reusable resource. So we came together, people in this Popsim consortium back in 2018. And this is really just a community-driven effort. There's nothing, we don't really have any funding. We're just people coming together, and it's really nice because we have pretty active Slack channel, and we meet every 2 weeks pretty much regularly. It's the only, I think, community effort I participate in that's really been continuously active and also productive. We'll show you some of the things we're able to come out, and all of you are welcome to join, first of all, and there are no entry fees. You just join, and it really is what?
Yay.
Yeah, but there's no food also, okay? So there's no, you don't get much, but very, very, very friendly and productive meetings every 2 weeks, where mostly we just talk about the things that people are currently doing, and we always have something that's kind of the next milestone in this. But as people join, sometimes the focus shifts, and we're now actually at a place where we're trying to think about the next things to do. I'll get to that maybe, hopefully have like a minute at the end to talk about that. So I think this is probably the most important slide in the whole talk, to just really promote this effort, because it's very important. And I'll show you, I have like 5 minutes to convince you why this is very important and useful, and then maybe talk a bit about the things we're thinking about doing later on. Right, so we have most of our formal contributions are on GitHub. It's just a code repository with a lot of simulations, pretty much, that are already encoded, and mostly people can just take the code and use it. It's designed to be very reusable and modular, so that you can combine different components. So again, now this is like the next generation in simulation. So if you had MS with this very cryptic code, not very usable, then you had things like MS Prime and Slim, where you could write your own documented Python scripts, which is much better. Now it's kind of even more abstract, because you just kind of take your species of interest, say what region you want, the demographic model you want. All of this is already coded. People coded kind of established models, and you can combine them, and then you run the simulations, only having to think about these kind of higher level things to select, and not the lower level details of how to implement the demographic model or model of selection, etc. Right, and you can do this in Python code, and you can do this in command line as well, and it's pretty simple. So as users, it's very, very easy. And if the simulation you are interested in is in the catalog, and the catalog is all documented, it's easy to look for things. If you find something that is under the scenario you care about, it's very easy to do. If you want to contribute, you want to add something more, then it's a bit more complicated, but not that much more. And again, the first and most important rule of standard Popsim is that everything that's in the catalog, it has to be a published, peer-reviewed estimates. So the idea is somebody published estimates none of it is truth. Right? The idea is none of this is the reality. We don't know what the population sizes were or the selection coefficients, but we want the things that are actually encoded to be based on a paper that somebody actually read and understood, where all of the methods are outlined of how they reached these estimates, so it's reproducible, and then you take these values and encode them in simulations. And the citation is part of the code as well. So everything is so each model that's encoded in Stdpopsim has the relevant citations for each of the kind of parameters we plug in there. And that's very important. That's one part, and the other part is this rigorous QC. So typically one person would code a model based on a published estimates, and then they would post it on GitHub, and somebody else would have to QC it, which is typically pretty much re-implementing the same thing and seeing that they fit. That's the baseline. Sometimes there's just code review because not everything you can actually compare, but even that, we almost always find issues. Like I think there are very few cases where somebody coded the model and it was just like very quickly QC'd. Always in the QC there's some discussion. Sometimes it's technical, somebody finds a bug, but sometimes it's conceptual things of how to interpret the things that are actually written in the paper. That's the big things. Now, one important take-home has to do with this simulation that I showed you earlier from that 2009 Gutenkunst paper. So as we were working on this and when encoding some of the models, one of the first models we wanted to put in was this model. So we took the MS Prime code that was written as part of the MS Prime paper to implement this Gutenkunst model, and we found a bug, right? So we found a bug by the people who really were the experts in encoding these simulations. It was a subtle bug. I think it's here. Yeah. So it's in one of these lines here. It's in one of it's hard to see, but it's just a simple line that turns off a migration rate. And that's a very common mistake in these simulations. And you can see that it actually has implications on things you'd actually see in your data. It's minor implications, so you won't obviously see it. But if your downstream inference really cares about these like edges, then that can be a big difference, right? So this was, and we keep seeing this. So no matter how good of an expert you are in coding these simulations, having someone else actually do the rigorous QC is super important. So I'm not going to say that there are no bugs in Stdpopsim. There probably are bugs, but it's, I think, significantly better than everything else. Just having two people, separate people, work on each of these simulations. Okay, yes, I'm pretty much out of time, so I will just mention the two. Won't have time to go too much into kind of recent progress, but we made two milestones since the initial publication of Stdpopsim in 2020. So one was to focus on expanding the catalog. So the first first generation of Stdpopsim focused on a few model organisms and kind of the most popular models that people used for human, Drosophila, and Arabidopsis, I think. And then the second stage was to expand the catalog for many more organisms and models. So we really recruited people from the community. And I think this is where a lot of the benefit will actually not be in human data. A lot of work is in genetics is in human data, but there the simulations are really very established. It's actually more interesting and important in other species. So that was one line of progress, and the other was to actually add a lot of natural selection. So the first generation of Stdpopsim were these neutral simulations using MS Prime, and now we also have we added ways of encoding these simulations with natural selection. So there you'd have to choose the regions that are under selection, and each region you'd have to have a distribution of fitness effects, and you can combine all of these now to create very realistic simulations of chromosomes where you have a detailed demographic model and pretty complicated and sophisticated kind of landscape of selection along the genome. And now this is really very realistic simulations of whole chromosomes, and you can do a lot of interesting things, which we do in this paper, which we published in bioRxiv earlier this year, and you can go to the paper to see. I don't have time, and if people want to do this, I may be just sorry, yeah. But I will just mention some things that we're working on now, because if you're interested to join, then you should know what are the things on the table. So I think the most exciting thing is to simulate traits, pretty much. So all of this is really just simulating genomes without any connection to phenotypes, and now we're trying to think of ways to expand to actually simulate traits, because that's obviously an important step forward. So if you're interested in that or have good ideas, then you're very much welcome to contribute. Another avenue that's interesting is simulation for training data sets for machine learning models. So there are a lot of work now in getting machine learning and deep learning methods to do a lot of these inferences, and for them you need to you typically have to have training data to train your models, right? And the more realistic your training data is, the better your inference methods are going to be. So it's kind of a similar idea where you have to, again, be able to simulate with a sophisticated demography and complex scenario of natural selection. So we want to expand to that and some other directions, and whatever people are interested and willing to actually do the work in, that's what we're going to do. So it really depends on the people who join. And acknowledge this is a partial list of the people who contributed, but I did mark in bold some of the lead authors on the 3 papers that we already published, and we hope to see you on this list for the next few years as well. Thank you.